they generate X-ray, and using a very concentrated beam of X-ray and pointing it at a crystal, you can actually study the structure of the protein, and it kind of works like this. So if you have, for example, a lysozyme, and you would grow a crystal of it, so crystallize it by, for example, drying it under the right type of conditions, you can, of these proteins you can get crystals, and then if you take the X-ray beam and shoot it at the crystal, the beam gets diffracted. It gets just like with a prism or with a lens, the beam gets diffracted, and this diffraction pattern, which is then captured, and often it looks like the picture on the right, that is then studied, and this pattern says something about the structure of the protein inside of the crystal. And there's a lot of tough math involved in this, because you have to kind of you have to calculate the angles at which all the X-ray beams are being diffracted, and how that relates to the internal structure of the crystal. But after the break, we'll go into optics and lenses and refraction, and diffraction. So I hope you then understand this also a little bit, how this can be done with X-rays.
This is called crystallography.
This is called X-ray crystallography. Yeah. Yeah, and this is also, for example, how the structure of DNA was discovered. They made a crystal of DNA and then shot the X-ray beam on it, and then from the diffraction pattern, Watson and Crick calculated and derived how the molecule was. That it has to be some kind of helix. Because a helix creates some kind of pattern of diffraction that is recognizable. Yeah.
Is that because that these parts are too small, that they can't be visually seen? What is the?
It's very difficult to see them. You can't see them with light because they're smaller than wavelengths of light. Therefore, it's impossible to see them with a light microscope. With the electron microscope, you can get a shorter wavelength by shooting electrons at a certain wavelength, and therefore, with a scanning electron microscope, you can see the outside of proteins and the landscape, let's say. But you can't really see what's inside of the protein with the electron microscope. Yeah. So, this is what I want to tell about, what the cell is made of and made out.